Okay, welcome back to chapter 16. So at this point in our story, we have gone through a bunch of different experiments, and finally the scientific community is in agreement that DNA is the hereditary material. And so now we are trying to study it to figure out everything we can learn about it. So the first thing that was going to be studied was how does its structure allow it to be hereditary? And so Maurice Wilkins and Rosalind Franklin, here's Rosalind Franklin, use this technique called X-ray crystallography. In X-ray crystallography, you make crystals of whatever material. So they made crystals of DNA, which is a very very careful, it took a lot of expertise to be able to even do that. And then took x-rays of them to see the shapes of the x-rays of the crystals and looked at the shadows and reflections they got back. And it produced this picture. I couldn't even begin to tell you how to read this picture. But this is the actual crystallography images that she got back. And because Rosalind Franklin was so good at what she was doing, she was able to look at this and say DNA is probably shaped as a double helix. This was one of her images that was swiped by, just straight up taken off of her desk by Watson and Crick, who ended up writing the paper and getting a Nobel Prize while she was left out of everything. So I think it's a horrible story of just ignoring people who are doing the work and other people getting credit when they shouldn't be. So I hate to give them credit when they acted like such jerks to her. But we're just going to say Rosalind Franklin did this work. Watson and Crick were really good at stealing stuff from her, taking credit for other people's work. That's kind of how I see this whole picture. So Franklin's X-ray crystallographic images of DNA enabled Watson to deduce that DNA was helical. And also looking at this, they could figure out how far apart each curve was from the next one. So they could figure out the width of the helix and the spacing of bases between them. And the shape suggested that it was made up of two strands, and that those two strands were forming a helix together. So then it was, if we're two strands, what is it? Is it T pairing with T? Is it G with G? So they tried some different ways. Are the bases on the inside or the bases on the outside? They modeled some different ways, and eventually this is the model that actually fit with the data that they had. That you had a backbone made of phosphates and sugars, and the bases coming in together. And you can see some of these bases are bigger and some of the bases are smaller. So if you'd had two T's together with each other, it would have been pretty narrow there. If you'd had two A's, it would have been too big. So you had to have a purine and a pyrimidine together, a small one and a big one together, to get that proper spacing. And using Chargaff's rules, they were able to put together, oh hey, purine and pyrimidine, A and T are equal, C and G are equal. That's probably because they're base pairing. So here is this double helix shape. So you've got that two strands twisting around each other. You can color these two blue backbones as this. That backbone is just the sugar and the phosphates. If you straightened it out, you'd have a picture like this. Here's your phosphate, sugar, phosphate, sugar, phosphate, sugar, and your bases together. T always pairing with A, G always pairing with C. If you turn these ribbons just into a ribbon on the side, you can just draw the bases out, T, A, G, C. And so these different ways, but what you might see is 5' to 3', 3' to 5'. So that's kind of important. If you remember back when we were talking about how you numbered the sugars around it, they're facing opposite directions, basically is what it comes down to. So while this is running this way, this one's running this way. So the strands are facing opposite directions even as they're pairing. And we'll come back and talk about what that means. Excuse me. We'll come back and talk about what that means in a little bit. So you don't have to exactly be able to tell me what a 5' or a 3' carbon is, but I do want you to know these two strands are facing opposite directions.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. A double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
So, Watson and Crick built models of a double helix to conform to the X-rays and chemistry of DNA. Franklin had already concluded there were two outer sugar phosphate backbones, and the nitrogenous bases were paired in the interior. And so these two strands are antiparallel. So I want you to think of like two lines of students. If you were in class, we could line up, and each line is facing opposite directions, but they're both made of the same thing. Here on this strand, this is a 5' to 3' strand. You can see their oxygens are facing up. Here, this is a 3' to 5' strand. Their oxygens are facing down. And so you always have these two strands that are running in opposite directions. That might not seem like a big deal, but it makes a lot of differences in how DNA is going to be copied because they're running in opposite directions. So please be comfortable with telling me one strand is 5' to 3' and the other strand is 3' to 5'. That means they are facing opposite directions, and enzymes are going to work on them in opposite directions. But they're both made of a sugar, a phosphate, and a base. Okay, at first, Watson and Crick got the bases paired like with like, so A with A and T with T, but that didn't give you a uniform width. So if you had two purines together, you got a model that was too wide for the data. If you had two pyrimidines together, you had a model that was too narrow for the data that they had. But if you paired a purine with a pyrimidine, that gave you the right width for the data that you had from that X-ray crystallography. And so A's and G's are going to be paired together, A's and T's are going to pair together, and C's and G's are going to pair together. So a purine with a pyrimidine give you the right uniform width. So Watson and Crick figured out that pairing was going to be specific and dictated by base structures. So adenine pairs only with thymine and guanine pairs only with cytosine. So this Watson and Crick model also explains Chargaff's rules that A equals T and G equals C. The relationship between their structure and function is going to be seen in the double helix. So because it's a double-stranded, a double helix, and they're specific base pairs, when it's time for DNA to copy, those two strands can come apart from each other and form base pairs to make two new strands. So here you have this old strand, and you can just add on the proper base pairing nucleotide at each strand so that you end up with this DNA able to base pair and make more DNA, this strand of DNA able to base pair and make DNA. I also want you to notice, remember, this strand is running 3' to 5', while this strand is running 5' to 3'. So our molecules are going to work differently. Here, this is a leading strand. The molecule is able to just run along in this 5' to 3', adding 5' to 3'. So it's able to run along. On this strand, this is a lagging strand. It's not able to run in this direction. It has to run backwards because it's facing backwards. So you're going to make a piece. Then you have to wait for DNA to open and make a piece. And you have to wait for DNA to open and then make a piece. And you end up with all these little pieces that you're going to have to put together. So the leading strand is made in one long strand, and the lagging strand is made in these little pieces. Each of these little pieces are called Okazaki fragments. So I'm going to let this run through, and while you watch what's happening here, I also suggest you watch that Amoeba Sisters DNA replication video at this point, so that you can get comfortable. We're going to talk about how DNA is copying itself. Okay, so since the two strands of DNA are complementary, Each strand is able to be a template for building a new strand. So here's your your two strands of DNA. So the base pairs complement with each other. So if you open them up, separate those two strands from each other, you can just base pair and fit in the missing bases in there. And then you end up with two strands. In DNA replication, these two strands unwind. Two new strands are built based on base pairing strands. And at the end, you end up with two strands that are both halfway made from the old DNA and halfway made from the new DNA. You don't have a totally old double helix and a totally new double helix. You have two double helices that are both half old and half new DNA. This is called a semi-conservative model. Semi-conservative model says that when at the end of DNA replication, let's see, you have two strands of DNA, each have one strand from the parent and one strand that is new. And so there were these other competing models. One was the conservative model, that you use that DNA and then copy a new strand, copy a whole new double helix. So you have one double helix that's totally old and one double helix that's totally new. That's not what happens, but before we did experiments, you didn't know that. And then one model was a dispersive model. It says just the whole thing breaks up and each strand has a little bit old and new and just all kind of dispersed and mixed up, which is also not what happens. But before we did the experiments, we didn't know that. So we did some experiments where we radioactively labeled the new DNA pieces that could be put in. So the old DNA is kind of this dark blue color, and the new is going to be this labeled lighter blue color. And what you see after one round of DNA replication is each strand is halfway radioactively labeled. So that knocked out the conservative model. And then when you copy it again, you have two strands that have that very original grandparent DNA now, and half of their newly made DNA. And then these two are going to be totally new because it's got a labeled strand here plus another labeled strand. So those results knocked out the dispersive model. So we understand that it's a semi-conservative model where each strand, each newly made double helix has a strand from the parent and a strand that was just made. So Messelson and Stahl, Matthew Messelson and Franklin Stahl, did this experiment where they talked about how they could figure out it was a semi-conservative model. And so this is the experiment that I just went through and explained, but let's go through it again. So you have this labeled nitrogen and a heavier nitrogen and a lighter nitrogen isotope. And then you centrifuge the DNA after a round of replication. You can see how heavy, how dense or less dense it is. So with the semi-conservative model, this fit the results we had after first replication, and it fit the results we had after the second round of replication, just by measuring how heavy each label was. And again, you can go through your textbook or rewatch this last bit to make sure you understand what is going on with Messelson and Stahl. Okay, the copying of DNA is very fast and very accurate. It's pretty remarkable how quickly it can work and how accurate it is. There's more than a dozen enzymes that are involved in this, and we're going to go through quite a bit of them, and you need to be able to tell me what each enzyme is and what it does. Other proteins also participate in DNA replication. Replication will begin at particular sites called origins of replication. An origin of replication is where the two DNA strands are going to be separated and you get these little bubbles. Here's an actual micrograph. You can see here's a strand of DNA and here's these two strands where the bubble is opening up, so you have this actual bubble. Here's another bubble. Here's a pretty big bubble where DNA is replicated a lot. A eukaryotic chromosome can have hundreds or thousands of origins of replication. Remember, we have very long chromosomes, and they can have multiple origins of replication, so it can get replicated and done. You're not waiting for one strand to come all the way through it. And replication proceeds in both directions until the entire molecule is copied. So here's the origin of replication, and we're going to start working in both directions on both strands. Here's this origin of replication. We're going to start working in both directions on both strands. Eventually, these two bubbles are going to meet up. These two bubbles are going to meet up, and you're going to have two complete daughter DNA molecules. At this point where DNA is actually being unwound and where replication is actually happening, is called the replication fork. Okay, let's watch this.
Replication of a DNA molecule begins with a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
So at the end of each replication bubble is a replication fork. This is kind of has this Y shape. This is where the new DNA strands are elongating. So the first enzyme we're going to deal with is helicase. Helicase is going to untwist the double helix and separate them. So helicase is coming along right here, just like is opening a zipper. So helicase is that first enzyme. Then we have these proteins called single-stranded binding proteins. Those single-stranded binding proteins are going to bind to single-stranded DNA. And that keeps that replication fork open so that those spaces don't just go right back to it. And then throughout this all, you don't want your DNA like twisting and getting big tangles and knots in it. So topoisomerase is going to help relieve that excessive twisting as things are being separated. It can break, swivel, and rejoin DNA strands, so it's going to keep the whole thing kind of from just turning into a big, coiled up, tangled mess. Okay, so three enzymes we talked about, or three proteins we talked about. This enzyme helicase, the single-stranded binding proteins, and this enzyme topoisomerase. You need to be able to tell me what each of those is doing. And if I ask you what enzyme does this, you can tell me. Or if I ask you what does this enzyme do, you can tell me. So know these enzymes backwards and forwards. Okay, then before we can start polymerizing DNA, we have to add a base. And so primase is an enzyme that is going to add this kind of primer to it to kind of get this whole thing started. However, primase is only able to add RNA primers. So these little primers that are added are made of RNA. We haven't yet started putting any DNA on it. Okay, so what does helicase do? What do single-stranded binding proteins do? What does topoisomerase do? And what does primase do? Okay, so we have separated those two strands. We have kept it open. We are keeping it from just like getting into a big tangled, wound up mess. And then we're adding a primer, that primer made of RNA. Then once we have this primer, it's going to build it, let's see. Well, let's talk about the primer first then. Primase can start an RNA chain from scratch, so it can just jump on, it doesn't have to attach to anything. And it's going to make these RNA primers by just base pairing with the DNA, putting the RNA pieces that base pair. Primer is going to be pretty short, maybe 5 to 10 nucleotides out of the thousands that the whole chromosome is. And this is at the 3' end of DNA, but 5' end of the newly made strand. And this is going to be the starting point where DNA can start being built. Okay, then DNA polymerase is going to add new DNA at the replication fork. So right here on this picture, once you have this primase has added the primer, then DNA polymerase is going to start adding DNA onto it. Most DNA polymerases require a primer and a DNA template strand. So for the DNA polymerase to bind, it has to have a primer to know where to bind, and it has to have DNA to bind to. And then it's just going to start adding DNA to each nucleotide. And it's going to add DNA at about 500 nucleotides per second in bacteria and about 50 nucleotides per second in human cells. So it's going really fast in the amount of time that we've been talking about this. A bacteria could be nearly done making its DNA, copying its chromosome. So DNA polymerase 3, so DNA polymerase 3, we're going to talk about a couple. DNA polymerase 3 is adding this DNA polymer to it. Each nucleotide that is added is a nucleoside triphosphate, and so you're going to take off those excessive phosphates to have the energy to do this. So DATP supplies adenine to DNA and is similar to how ATP works. It's regular ATP. Although here DATP is a deoxyribose, so it's the DNA version of ATP. And as each monomer joins the DNA strand, we are going to have a dehydration reaction. So water is released to combine two pieces, and it will lose its phosphate groups as a molecule of pyrophosphate. So these nucleotides that are being added have phosphates on them, and we're going to just cleave those phosphates off to release energy to be able to grow the DNA. Because DNA is antiparallel, remember they're facing in opposite directions, this is going to affect how DNA is being replicated. So here, if this is a primer, DNA polymerase is able to function just following along in this direction, following right along behind the replication fork. However, on this strand, because it's backwards, DNA polymerase can't follow along the replication strand. So it's going to have to make a piece, then come back and catch up, make a piece, come back and catch up, make a piece. Okay, so this is going to be the leading strand, and this is going to be the lagging strand. This side of the origin of replication, this is the lagging strand, and this is the leading strand. Okay, so leading strand means where the DNA polymerase can just ride along and make one whole long piece. The lagging strand is a section that is always kind of waiting to catch up. It's kind of lagging behind, waiting to catch up. It has to build a new primer and build new DNA again and again and again as DNA is unwound. So DNA strand can elongate only in the 5' to 3' direction. It can't function running both ways. So the leading strand is a strand that is made continuously. It's just made in one continuous piece. It's always moving towards the replication fork. Let's look at the leading strand.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The two strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
Okay. For the other strand is the lagging strand. So it has to run in this direction away from the replication fork. So it can't just follow along. So it's going to wait for it to open a little bit, put on a primer, and run. Wait for it to open a bit, put on a primer, and run. Open a bit, put on a primer, and run. That leaves you with these little pieces called Okazaki fragments. And then when you have a lot of Okazaki fragments, you don't want to just have DNA made in a bunch of pieces. So we are going to seal them together with DNA ligase. So DNA ligase is a little gluing enzyme that is going to connect each little primer or each little Okazaki fragment together.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA.
So it said another kind of DNA polymerase. So first we had DNA polymerase 3 that made the DNA. It's DNA polymerase 1 that will take out the primers.
Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand.
Okay, so you should be able to tell me how the leading strand is made, how the lagging strand is made. You should be able to tell me what each of those enzymes are doing. What is topoisomerase doing? What is helicase doing? What are single-stranded binding proteins? What is DNA polymerase 1? What is DNA polymerase 3? What is RNA polymerase? What is ligase? What is the difference between a leading strand and a lagging strand? What's an Okazaki fragment? Those are all things that you need to be able to know from memory for this next quiz and test. So let's overview this, and then this will be the end of the part 2 video.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles, which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding 2 identical DNA molecules.
Okay, so I will meet you back for chapter 16, part 3 lecture, where we are going to finish this chapter. And let me know if you have any questions. I'll see you soon.